Okay, 3:05, I think it's time to get started. So welcome to everyone to this seminar of the series we hear from ECLT. It is a really great pleasure for me to introduce Professor Doron Lancet, of course, from Weizmann Institute. Doron, of course, needs no introductions to this community because nor to any, the international community, because it is well known. He has been with the ECLT network from the very beginning. But I'm just doing this for the record because this has been recorded. So let me do this. So Doron was trained first at the Hebrew University in Jerusalem, and then at the Weizmann Institute. And then, as often the case from people, graduating from Weizmann, he moved to US, more specifically to Harvard and then Yale, until eventually he returned to Weizmann to become a faculty there, and he has been ever since a professor ever since 1993. So among many contributions that Doron gave to the international community, he pioneered this idea of a novel lipid world, in which I think he has become a leader, international leader, and I think he's going to share us the fact that he has solved the problem. Somewhat that's what he was pointing to me, few minutes ago, and we are all eager to hear this. So without further ado, let me give the floor to Doron, thanking him again for having accepted our invitations. Doron, please.
Thank you, Achille. It's a pleasure to be in Venice again. I'm sorry I am not in my physique, but I'm here. I'm with you with soul. I will speak today about a few aspects of lipid world that have to do with innovation on the major theme, and a few points in which we get more convinced that we may be on the right track. So the title is Self-Reproducing Catalytic Micelles as Early Nanoscopic Protocells. Most people think they know what a protocell here is. You see on the right bottom here, this is one picture that I found when I asked Google Images to tell me what a protocell is. I usually go to Google Image to ask questions. It's very good answers. Each one is 1,000 words. And this is one picture I saw. This is a protocell dividing, and not clear exactly what is inside and what kind of glass it's made of. But this is a protocell. Actually, nobody knows what a protocell is. But there are a few definitions. And one good definition is that protocell is before LUCA. It's much simpler, but that it can do a lot of things. That is, it has, a lot of people think, some RNA, some proteins, and so on. So it's a long way from the prebiotic soup. And we're trying to find some paths that will facilitate going into what people believe are protocells. What are micelles? Well, vesicles or liposomes, most people know. These are these guys here on the left top. And this is a bilayer that everybody knows, but this is a micelle. A micelle is actually a monolayer that became a sphere. And for those who are physicists or philosophers, the white ones are water-loving parts of the chemistry of a lipid, and the orange ones are water-hating. And in a miraculous way, if you add such a compound to water, not always it's very easy, but if you do, you get all of these kinds of structures. A bilayer is most of the time also spherical, but much bigger than what is shown here for liposome. So the cells around our membranes are also bilayers like this. So we are going to actually speak only on micelles because we got convinced that there are so many advantages to start life with micelles that nobody will resist that. Liposomes are wonderful, but we think that liposomes or vesicles are already a higher stage of life. So we try to begin with the most rudimentary, and size is one. The typical micelle is 10 nanometers in size, and a typical vesicle is 1 micrometer. So it will be like a vesicle is 1,000,000 times in volume than a micelle. And this small size, the nanoscopic size, is an extreme advantage, which I'll tell you more about. So what is lipid world? First, we have to ask, are there lipids in a prebiotic soup? And the answer is yes. It's been around for many, many years. 1985, the famous paper by David Deamer that showed that extracts of the Murchison carbonaceous chondrite meteorite contained vesicle-forming compounds. But if you go to the middle here, this is done by collaborators of us in this paper. This collaborator, Philip Schmidt-Koplin from Munich in Germany, has been doing for hundreds of different particles that come from space, mostly meteorites. What is the total chemistry? Not just asking, is there valine there, or is it a little bit more L-valine than D-valine? He asks what is there, and he gets hundreds of thousands of molecules that are in each of those meteorites. And many of them, as shown in this diagram, in which from left to right, it is how many carbons, and from bottom to top is how many oxygens. You see that there is a very good predominance in one of the fractions that he extracted with different solvents. This means that very few oxygens and quite a few carbons, which means an amphiphil most of the time, depending on the structure. So I also show, this is in a paper that we published about some analysis of Enceladus, the subglacial ocean of Enceladus, the moon of Saturn. And there is a wonderful ocean there in temperatures between 100, maybe a little more than 100 and somewhat less, but still liquid water. And there are organics, as we know from the mission of Cassini and the work by Postberg and others that was published 2 years ago, 3 years ago. There are a lot of organics. You know, people go to Mars and say, well, the organics. If you go to Enceladus, you don't have to ask that. There is a lot of organics in the geysers that go up, which Cassini was able to analyze in space. And this is all published. And many of those compounds are very complex, and they are of the kind that if heated by pyrolysis would give rise to a lot of small, nice compounds. And the solids in those droplets that come from Enceladus are very similar to what is in a meteorite, like Murchison. So organics are all about, we don't have to synthesize them. We don't need Miller and Urey. They come out of space all the time, much more higher flux in early days, less so today, but organics are not the problem, and lipids are not the problem. So one advantage for vesicles, I have to be polite. Vesicles have an inner space, a lumen, which contains water, and all around there is a bilayer. Micelles don't have a lumen to speak of, but actually they do. If you read the literature, you realize that, okay, these are all water soluble inside here. They cannot be lipophilic, they cannot be amphiphilic. But if you look at a micelle can absorb almost any molecule. If it's hydrophilic, it stays on the surface here, but quite tightly absorbed in many cases. And if it is halfway, it goes deeper and deeper. And if it's very, very hydrophobic, it goes in the core here. And if there is a lot of this substance inside, it becomes an emulsion droplet, which is a big brother of a micelle. A lot of compounds can go in, and importantly, while you cannot, unless you have some relatively advanced machinery, biochemical machinery, you cannot have concentration. Enhancing the concentration inside doesn't really work unless there is absorption to the inner side of the membrane, which is a little bit like what happens with a micelle. But micelles can concentrate external compounds by factors of 10, 100, 1,000, or even more. And generally speaking, because there is no water, this particle, any molecule that is in this particle, has a huge concentration, sometimes up to 1 molar if you compute. So all of these problems of dilution, of how molecules will find each other and so on, all of these questions go away when you believe that micelles started everything. Now, maybe I should say one more thing, and that is that the outer surface of a micelle is like an outer surface of a bilayer membrane. And you can get absorption or even non-covalent attachment to the head groups, the hydrophilic groups all around the micelle. And Günther Blobel, a famous, very famous membranologist who worked on transport of proteins across the membrane and a lot of other things. He got the prize in 1999, and he suggested in one of his earlier papers, actually mid-papers, he suggested that in very early life, functional molecules were located on the outside of membrane assemblies, as shown here. And then he goes to an interesting story how it became a double bilayer and it closed. And by the end of the day, some of the molecules find themselves inside. But the main message that I'm trying to convey is that the outside is a very good business location for a lot of interactions. And all of that will be part of my talk today. But I also have support from a Nobel laureate, which is nice. I have a few Nobel laureates that don't support me, such as Jack Szostak, whom I adore and like, and we're good friends. And he was a mentor of my second PhD student who went there for a postdoc. So we know each other very well, but he doesn't like my model. He works on lipid a lot, but lipids as barriers, as compartments. We say that lipids can do a lot more than just being barriers. Now, lipid micelles can reproduce autocatalytically. This we have known for 30 years now. Louisi and colleagues have done the pioneering experiment, and since then it was done by many others in different variations. And the idea is as follows. If you start here, the red guy can be an anhydride or an ester, so it's very lipophilic. It's not soluble in water. It makes an oil slick on the water or a droplet, and if it hydrolyzes, which it does very slowly in water, it gives rise to that, which gives rise to micelles. But once the micelles are there, suddenly there is an enhancement of 100-fold of how fast this gets hydrolyzed. So this is autocatalytic formation of the building block of a micelle. And for that, the kinetics goes that as long as its background rate of hydrolysis, it goes like that. And then once micelles, once the solute, the green solute goes above critical micelle concentration, suddenly there are micelles, and suddenly there are many, many more micelles. This is seen by light scattering or some other methods. And many years later, Stephen Fletcher has been working at Oxford, with whom we interact a lot. And he published many papers on this system. In this case, with a small difference, that a lot of what he does has to do with tails in an organic layer, heads in aqueous layer. They have sufficient chemistry to talk to each other and form a covalent bond. A monomer forms. Then after this is filled, it begins to form micelles, and the rest is the same history as with Luigi Luis. But it turns out that what micelles can do is a lot more than just making their own building blocks. They can catalyze many, many reactions. The reason most of the people in Origin of Life are not absolutely aware of that is that most of the papers in which this is published, some are in JACS and so on, but many are in industrial literature. But industry likes micelles a lot. By the way, I have to say that whenever you wash your hands with soap, your hands are just coated with micelles, because this is what removes the oil and the dirt from your hands, because it's such a good absorber, and it absorbs what pure water cannot. So the idea is that in industry, they use this as a replacement for doing synthesis in organic solvents, because if the synthesis happens inside the core of the micelle, then you don't have a solvent, you don't poison the environment, and you get very efficient synthesis. So there are hundreds of papers that show that that micelles can be catalytic. Of course, they should have head groups of the right kind. As you can see here, this is epoxy, and this is a trimethylamine for positive charge. Here there is just a proline, which is very, very modest molecule that is supposed to be somewhat prebiotic at least, but I don't care about that so much because in prebiotics, apparently almost everything was there. But I don't tell people because they will shout at me and hate me. So basically, if you have proline here, it catalyzes this reaction by what we call trans. It doesn't get back to the lipid or to the bilayer or monolayer, sorry, but it just is a catalyst. And here, what you see is that peptide bonds are synthesized and they become attached. So you can generate this way a peptide attached to a head group or being the head group. In this case, SH group is important for those who are chemists and so on. But all of that just says that micelles are extremely powerful as chemistry power plants. Now you can do much more in life today. For example, this is a lipid with two tails and with a diphosphate nucleotide here. Actually, it's, you see, this is diphosphate, this is, what is it? Cytosine or something like that. And so it's a lipid with a base, a DNA base or RNA base. And this is a cyclic peptide that is attached by some bacterial species to the tail. So you can get almost anything. This is a very important take-home message. A lipid is not what you see today as lipid. A lipid is anything that has a tail and has a head that is like that is hydrophilic. And you can get a lot of diverse chemistries this way. And if you start with an ocean that is very diverse to begin with, then you'll get lipids of all sorts of stuff attached to them. And also the tails can be very, very different, small or large, with or without a double bond, and so on and so forth, with branching or not. So you get a lot of diversity. Now, how can a fluid do something important, such as bind non-covalently a polypeptide? If you wish to say that lipid micelles started it all, you have to sort of also ask questions not only about covalent bonds, but on non-covalent bonds. And this is a paper that we are now writing. I should say that a lot of what I'm telling you about is a very talented master student, Amit Kahana. He was mentioned in the first slide. And this is part of what he's doing. He's now working on his 6th paper as a master's student, which is quite unusual. So the idea is that something that we are working on with some other people, something that we call a dynamic uptemer. And that is that although the micelle is fluid, temporarily there can be a combination, say, of their green, purple, yellow, and blue in the vicinity of each other. And once they are there, something can bind. So this is an important, I will not go into detail, but it's an important story. And in real biology, we have just that. There are proteins that interact very specifically with bilayers, with certain compositions, and they do so with, you see these little ones, where are they? The blue ones here and here are attached to the protein, and they go as if they were lipid to help the protein attach. This is caveolin that makes it makes a particular membrane structures that lead to vesicle formation from the plasma membrane. So anyway, the idea is that this can happen. Now, let me speak about a few advantages in addition to what I said already, and I will at the end say even more. Small size, I already mentioned, for very, very high concentrations, because actually it's the same concentration as in the bilayer if something absorbs here, but they are better absorbers and much more diverse. They form much more readily because of chemical promiscuity. So if you start making micelles, you know, there are many papers in the Origin of Life. You cannot get vesicles if the concentration of calcium is higher than something, or if the pH is too small, and so on. The micelles don't care. If something outside is wrong for one type of micelle, it will be good for some another type of micelle. So different compositions of micelles will form in different environments, but you don't have to wait forever until the conditions are just right in some God-forlorn a hot spring that has just the right concentrations to make a vesicle. Here, it will happen everywhere. So to me, the question of where life began on Earth is immaterial. I'm sorry I'm saying that because I know, again, I may offend a lot of people, including very good friends. But what really is important is what started life chemically and what were the initial steps towards something that might look like a protocell. Okay, so promiscuity is extremely important. There is a very high surface to volume ratio if you're very small, so that's important. Greatly enhanced concentrations, I already said that. Augmented diffusional interactions because it's a fluid. Polarity gradient, I mentioned that. And it looks a little bit like what a lot of people in Origin of Life use, that is minerals for catalysis or proteins for catalysis. Proteins were not made yet, at least not very easily. And minerals were abound around, but you cannot take them with you. An entity that lives has to be made of organic compounds within each entity. There should be all that is needed for it to survive, to catalyze, and to divide, and to mutate, and to evolve. And you don't want to have a grain of mineral taken around, although in our body we have nice minerals like teeth and so on and bones. Okay, so let me go on. These are some of the advantages. Let's jump a little bit to the ideas that 50 and more years ago, Stuart Kauffman propagated. Most people, again, don't believe him either, but we are on the same side of the wall. He said something very simple. If you have a lot of different compounds, and if one or another catalyzes a reaction between one or another compounds in that set, then if you have sufficient types of molecules, the probability of catalysis will increase. And then by the end of the day, you get what is written here, catalytic closure, which underlies reproduction. We have done a lot of work to define in our micellar systems whether or not they are catalytically closed and under what conditions, and we found the answers. And we also went some steps further than saying if everybody catalyze, if every molecule can be produced by a reaction catalyzed by another molecule in a set, we took it further to say what exactly is necessary for actual reproduction to occur, and I'll come back to that in a moment. Now, even if you look at that, there are molecules, these are peptides made of two types of monomers, A and B, different sequences, and where they are, we never heard from Stuart. They're there. And to me, it's very clear that if they need to do what they do, they have to be together in a defined entity. And that defined entity can be a micelle, because then the molecules are very near to each other. The chemistry is in the heads. The tails keep them together. The tails are very non-specific, so you don't care about what keeps you together. But what is on the protruding on the surface, that is very important. And that is what can give rise to catalytic closure. So we feel that we are standing here on the shoulders of the giant, Stuart Kauffman, and we help, like the person who said that, I'm sorry to say, I shouldn't say that, but the person who said I was standing on the shoulders of giant was Isaac Newton. And he said that he learned a lot from others. I don't say I'm Newton, but I say I learned a lot from others. So can you actually see papers that define or show mutually catalytic networks, as those are, or collectively autocatalytic sets, as Stuart often calls them, CAS, sorry. So yes, the answer is yes. A good colleague of mine, Neil Devaraj at UCSD in San Diego, has been doing this work for many years now, for almost a decade. And he shows wonderful networks, as shown here. No details, but here there is a catalyst that contains copper, but that's okay. A lipid can be a chelator for a metal in its head group. So that's absolutely okay. And there is a model by Hodike that is called RAF, and we show that under his definition, I should call it computer science definition of what is a good mutually catalytic network, yes, what the Varge did is a mutually catalytic network. And this is published in a paper that we published in 2018, a big review in the Royal Society interface. Okay, so let's continue. I started 5 minutes after, right? And I should end 45 minutes after. Is that correct?
Yes, correct.
Okay, I'll have to make an effort to do that. So, revisiting what you see here in the picture, what actually reproduces when these green lipids grow and split, lipid micelles grow and split? The answer is sort of nothing, because everybody is green. But what if it is a mixed micelle? Then a completely new era dawned. This is extremely important. That is, now you can ask whether this guy here, the daughter and the mother here, before it grew, whether they are the same composition. So composition is actually the denominator which is used for micellar origin of life. There is no sequence. There are definition of molecules, but no sequence. So composition plays the role. And what is composition? Just how many of each kind. It's a vector. It's very easy. You can count how many reds, 3, how many yellows, 7, and so on. It's a vector of composition. And you can ask also how similar what is linear sequence-based composition, information in an RNA molecule versus what happens in a lipid micelle. And the answer is that they both contain information. And actually, as the molecular diversity, the alphabet goes up, they are asymptotically similar in terms of what size of entity shown here is necessary to get 100 bits of information, of Shannon information. And there is a formula also for showing how much information. But this is not the end of the story of compositional information. Cells mostly transmit compositional information from one generation to another. How many of each RNA kind, what is called the transcriptome? How many of each protein, which is called the proteome? If these change, the cell dies. So they have to be very careful about growing while keeping your composition put. And the same either for proteins or for metabolites or for RNA and DNA. DNA is not a story because it's the same concentration, 2. But in the daughters, there is also 2 in most cases, 2 copies. But others have a very diverse compositional space. And cells, when they grow up and split, actually use their metabolism, broadly defined, including the RNA synthesis, the DNA synthesis, the protein synthesis, and so on. They do what is necessary so that after mitosis, they grow, they keep the concentrations the same, and then when they split, as shown here, they split and hope to have the same composition. Now, of course, random lipid micelles will not breed true. But what we discovered by computer simulations that I will describe in a few moments is that they can do so if they have the right composition for that. But that is also true for a living cell. It will not do anything unless it's in the right composition. So that's nice. Okay. Now, we now know that lipids and RNA both harbor information as well as catalyze or involve catalysis. So which is better? The answer is, in addition to many other reasons, is the following. For RNA, you have to do a lot of filtering or synthesis that ignores all the other molecules, which is not very easy to understand, but never mind. So you have to come to the right molecules to make RNA or proteins. But micelles don't care. I also said it many times. They can spontaneously form in a very dirty soup. And as I will immediately talk about, they can be of different compositions. Some are good and some are bad. So to make that out of this is a total improbability. Most people agree to that. But when they say that, they say, we have no other choice. This is the other choice. So that's the model. No time to tell about it. And also I told about it, I think 2 years ago or 3 years ago. and at ECLT, and I spoke about it many, many times. But there is a set of equations, you can simulate them. What is important is that you end up with all of those, and this of course simulates a non-equilibrium process, which is important because life is non-equilibrium. And we published this in many, many papers. But the idea, let me jump just a second to here. What is really important is that this process is shown by the simulations to occur in such a way that there is a lot of lipids here, and they begin to grow a new lipid micelle, make it larger and larger, and then the split is just a physical phenomenon that we know happens. We don't care about it. But this has to be homeostatic growth, preservation of relative concentrations. And certain compounds do that, is shown here. The certain compositions of compounds do that. They roam in compositional space. This is one compound, second compound, third compound, and it's actually in our simulations 100-dimensional. But they go here and they stay here for a relatively long time, which means that there is growth and split without changing composition, which is exactly what you need to start life. What is needed to reproduce. Now, of course, if you are a vesicle, as pointed out by Roberto Serra, by Sole, and so on, other fellow, you have to actually connect between a Kaufman-like reaction network inside and what happens in the lipid bilayer. Roberto calls it a synchronization. We call it a coupling of the molecular reactions that happen here with here, but it is exactly the same. Let me skip that because I don't have enough time, and just say that there is experimental evidence for homostatic growth in mixed lipid micelles from the lab of Fletcher. And I will not go into too many details, mainly to say it's a flow reactor, so it's away from equilibrium. There are 3 different tails with 3 different colors. There is one type of head, and you end up with concentrations of the 3 different lipids formed, as already described before, with different concentrations, and they keep as long as the flow goes, which means it's a steady state with homeostasis. I will just say, without explaining anything, that we are now doing molecular dynamics also, where we show how this molecule monomer goes into a forming micelle, and we ask questions about how the composition here affects the kinetics of in and out, which is extremely important for our guard model. No time for detail. So, as we went with this guard model, we discovered 2 interesting things. One is that composomes are dynamic attractors. And what does that mean? I know there are many people in the audience who know much more about attractors than I do. But the basic idea is that you have a space shown here, component X, component Y, component Z, if it's a compositional one, and there are trajectories. And the basic idea is that attractor attracts, which means that wherever you are in space, in this compositional space, there is a tendency to go here more than to go there. And if you're here, if there is a perturbation, you tend to go back to that. And it's difficult to emerge out. That's an attractor. But Amit did a lot of guard simulations with the idea in mind that he can show whether or not, if you start from different compositions, different random compositions in different colors here, in advanced guard simulations, this is composition similarity. This is a phase diagram as it's known. I don't have time for that also, but compositional similarity to compotype, which is like the composome, and compositional similarity to flux. What is important to make life out of micelles is that the flux in terms of how many of each kind, will be proportional to what is already inside. That's what will give rise to homeostasis. And lo and behold, start from many different kinds. Let's ignore this for a moment. It's a by story, but many of these go and converge in this particular point, which is showing that there is an attractor behavior. We have some other evidence for that, but never mind. So what we can say is something as follows. Here we know that there are many, many different compounds, and the micelles are very small. Under these conditions, you can get a lot of different compositions, and it's like a a combinatorial library, and there can be a pre-evolution. This will go to the attractor and this not. So we do a lot, we do not millions and not billions. We computed that, and it can be 10 to the 30 different micelles in the first depth of 10 centimeters all around Earth. So we look at it as a planetary phenomenon. There are many, many, many different kinds of micelles, and some of them will be better than others in starting the journey towards a composome in which they actually are breeding through, but some will. So to sum up, origin of life in the lipid world, we say the following. You start with a library. There is a chance to get something that by some effectiveness or another will be able to catalyze this. I didn't say very well, so let me say that. The homeostasis is caused by the fact that what is already in catalyzes to different degrees what aspires to go in. And this gives rise, under some conditions, to flux equals in compositional direction to what is already inside, which is homeostatic growth. So many of those won't do that, but this one perhaps will. Then it grows, then it's split. Split is noisy. They are not the same here because there is a statistical noise. And this is a bit like compositional mutations. There can also be compositional mutations on the way up, as well as on the way down. Now, this, of course, is a mystery and sort of remains a mystery. But that is, how do you go to a more elaborate, more evolved vesicular protocell? But if it's true that the micelles breed through, if it's true that there are mutations, if it's true that there is variation of micelles, as Darwin asks us to have a selection, to have natural selection, then it looks as if micelles can begin the job as small as they are, as simple as they are, and as readily forming as they are. They can start a very, very long chain of evolution. But the evolution should always be with an entity that breeds through, or else everything is broken down. So you have to think about what happens between this and that. There can be changes of chemistry, like a second chain tail added to cause vesiculation. There can be changes of the size of the head and so on and so forth. But basically, one of the steps is to go to become vesicles, because that is what life today is. But also, the chemistry can become much and much better, because the attractor actually spews out a lot of the compounds and leaves a few behind. So only a few out of the gemisch outside will make the micelles. And this process can go on and on and on for hundreds of millions of years. And since we know that evolution, I'm sorry to say that it's a bit of far-fetched, but as we know that by evolution you can go from a bacterium to an elephant or even to a human being, that is not a complete miracle. That if there is already selection, survival of the fittest, and complexification, which is part of evolution, then you can get that, and then you can go all the way, maybe in another 500 million years, to what we call LUCA, which means that it has a ribosome, it has metabolism, a very well-behaved metabolism, it has RNA that can encode, it can proteins that can catalyze much better than micellar lipids. But this is just the same as saying that the elephant thinks much better than the bacterium. This is the end of the beginning. I think I'm on time, and I'll be happy to hear questions and also criticism, of course.
Thank you, Doron. This is a virtual clap that I'm making on behalf of everyone. So, are there any questions? I guess that you are going. We're going to see your. I spotted that you are going to publish this soon, so probably we can read this more, right?
Okay, 90% of what is here, I would say, is already published in many papers.
Okay.
There is a paper that is almost accepted in Nature Review Chemistry, in which we tell the story of micelles in much detail. Mm-hmm. I don't worry about. I talk to you, and you can actually do whatever you want with it.
Yes.
And the attractor story is still in writing, and the molecular dynamics is still in writing, but I never covered up any of my work before it's published.
Okay. Claudio, you have a questions? You know, you flashed, there was a...
No, it was not a raised hand, it was just clapping.
Oh, okay, it was clapping, sorry. Are there any questions from the Living Technology bunch? I guess that Roberto, maybe Roberto is around, maybe he has questions. I'm sure that, yes, Roberto is around, probably he has questions. I have many questions.
If there are no questions, that it means that they all agree, so it's okay.
Yes, no, there are questions. Martin, the first question is by Martin. Please, Martin.
Okay, thank you, Doron, for that presentation. It's wonderful to see, as usual. A question, just kind of a technical question. So let's say you're able to produce these composomes. What I remember from some of your papers is they persist, but they do have a limited lifespan, don't they? I mean, lifespan, let's say. I mean, how long do these things need to persist? They're not going to stay as a composome forever, presumably, right?
Okay, it's an interesting question, because I encounter a lot of people that ask me about stability. And it looks like there are a lot of papers that are about stability. That is, RNA is not stable. That's one of the critics' opinion about it, that it cannot start life because it's not stable. Micelles or vesicles can be more stable or less stable in one way or another in different conditions. Life is not stable. It's not supposed to be stable. It's supposed to have a viable steady state. Okay, so asking the question of how long it lives is interesting, but it depends on the speed of the processing. If, for example, just take into account the possibility that micelles form and decompose or split, let's say within a minute or something like that, just as an example. All you need to do is to make sure, and also what is stability? Stability is the composition. The composition will change all the time. Molecules will come in and come out. But because there are some, and I didn't say that very clearly, but before we start with covalent bonds, our GARD model is basically non-covalent. So molecules go in and out, and some go in much faster than others because the entry is catalyzed by molecules inside. So all depends on, there is a competition here. The competition is between what may make noise kill everything and what is important to make the next generation. If you make the next generation, then you don't care. Maybe one of them will decompose and the other will not, and so on. So this is just exactly like Darwinian life processes. You can live one day, or like a butterfly, or you can live 100 years like a turtle, but you make do. So please ask me if you want more specifically, what would you like to know about stability? I thought
I'm not interested so much in stability, but I guess what you're saying is it's a succession of different types of composomes. Is that what you're saying?
No, what happens is that if you are in a status of composome, then the kinetics keeps you intact. That is, you end up with a process which is like the attractor. If you deviate from the attractor, it pushes you back. So this reactivity of molecules inside the micelle upon the entry of other molecules from the outside, because it has an attractor behavior, will make it less easy, let me say it very carefully, less easy for the composome to completely disappear. In our simulations, we have some composomes in some experiments that live for 20 growth split cycles, and there can be more than one composome for a given system, so there can be another composome that is much more short-lived. But this is exactly what evolution is all about. So I don't usually ask how stable is a rabbit. I ask whether it will make enough offspring before it dies. And that is apparently what you also have to ask about the to the composome.
I'm confused a bit because if you're just considering attractor states, where's the evolution?
No, it takes care. Attractor doesn't take care of evolution.
No.
Attractor is just making sure it happens. Nobody directs it to do so. It happens by solving the equations. But what happens is that it makes sure that that some of the progeny will survive. If we run this simulation, the composomes never die. We do also simulations in a flow cell. Okay, so we have 1,000 micelles inside. Some in one composomal state, some in another, and some are not very composomal at all. Okay, and the flow cell takes micelles out and brings monomers in, and it can work forever, like life works forever so far since it started. So I'm sorry, it's really an extremely important point, Martin, really, but maybe I didn't give enough. Maybe I think that because of this good question, I will try very hard in our next paper to address it in the best possible way. So I really thank you. If you still feel that you don't understand, since there aren't too many people wanting to ask, you can ask a little more.
Yes.
I'll leave the floor to the others. Thank you, Doron.
Thank you. I should mention that Martin and I speak very often and share ideas and thoughts about how to prove composomes soon, but it's very nice, Martin, that you ask a devil's advocate question. It's extremely important.
I see that Maria has a question. Please, Maria.
Okay. Oh yes, thank you. First of all, thank you so much for this fascinating talk. There are a lot of new topics, but I was able to follow the general idea. It was really engaging and clear. So I was just wondering about one thing. So what is the role of computational research here with mathematical models? Because I saw here and there a lot of references to mathematical concepts and ideas. So for example, when there was the hypothesis of early life with functional molecules outside and then get inside and so on. Also the role of some kind of formal grammars in catalytic closure, and of course the role of attractors. So I'm wondering about the potentialities of computational and mathematical modeling and the limits here.
Many thanks for this, Maria. That's a very good question. When I go around in conferences, what I used to be until recently, going to conferences, people always say, this is the guy with a model. How can you prove this model? This is just an invention, a figment of your imagination. And I say, no, it's not. So there are many, many models for life. And most of them don't have equations. And if they have equations, the modeler decides about the parameters, the functionalities, the constants, the variables. We usually call them toy models. This is not a toy model. Let me say a few words about it. Where am I? Can you still see my screen?
Yes, we can.
Okay, so let me see. So let's look at the equations for a moment, and I'll say a few words that I did not say beforehand. Oops, here. Oops. Okay. So first of all, it's a kinetic equation. So it's like following enzyme with substrate, like Micelles-Menten or something like that. Okay. And there is Ni, that is one of the compounds. N is how many of them? There are constants, rate constants. This is the total number of all molecules. And that is extremely important. Okay, first, the change of Ni is proportional. There is a basal rate, which is these two forward and backward times 1, plus a term that is multiplied by a sum of beta ij. Beta ij are how much catalysis molecule I exerts on the entry, catalyzed entry of molecule J. Now, a lot of people would just throw a random dice to populate this beta ij. We did not do it randomly. Sometime before I started doing work on origin of life, and actually what led me to this molecular scrutiny of origin of life, I wrote a paper, a couple of papers that have to do with the propensity of high or low affinity between randomly encountering molecules. For example, a huge library of ligands and a receptor, a huge library of antigens and an antibody, and so on, or drugs and protein drug targets. There is a lot of information on that out in the literature. And we discovered that the probability of high or low affinity, or the probability of high or low catalytic effect, are log normally distributed. And we also have more or less an idea of where the numbers are. And we stick into the model, this beta ij, not by guesswork, but by basing it on real chemistry. And the difference between GARD and many other models is that it attempts as much as possible to rely on real chemistry. The equations are chemistry, the laws are chemistry, like away from equilibrium, delta G, etc., etc. All of that is total chemistry. It's not a, what did you call it? A formal grammar. It's not a formal grammar. Absolutely not. Have I answered, Maria?
Yes, thank you so much.
Okay, thank you. John also has a question, please, John.
Okay, just managed to unmute. Doron, it's a delight after all of these years to witness the fact that the composome of your presentation is still alive and ticking, and so preserving its composition to a significant extent, so that one can still recognize it as the same composome as at least 10 years ago. So that's one example of a verification of your theory that the composome of your talk is surviving. But the issue that I have, which is more a question than an issue.
By the way, I just have to say that that this survival, our daring to write more papers, let's say in the last 5 years, has to do with a revolution in the last 5 years in experimentation that is not just has a far resemblance to our model, but actually they cite our model as the reason for doing the experiments, and the results is proving some aspects of the model.
Yes, I'm impressed. I was not intending to say that, to imply that that nothing has changed. I'm just trying to say that it's that the recognizability of your presentation is still there. So that there's continuity is a good thing. Another a sort of peripheral continuity, since we're going on that point a little bit, is just that the choice of colors for your different lipids, Smarty-like, has also survived, whereas one might have thought that was a phenomena that would not survive the course of time. So there are some nice sort of spangles of St. Mark's type things in your presentation that indicate that even non-intended artifacts are surviving. But listen, the main point I wanted to make is about it's somewhat related to Martin's question, but not totally. It's about orthogonality. And as you, of course, understand these things, but with genetically based, nucleic acid-based replication, there is a fundamental orthogonality in the structural means of replication and the variation in properties, which is significant. So that, to put it simply, if you come up with a new sequential mutant, a variant, which does something better, then the task of copying that variant is not a new problem that has to be resolved. It is an orthogonal property of the whole family of RNA molecules that they can all be replicated, perhaps at different rates.
What is it perpendicular, which orthogonal means to? I don't understand. I understand what you said, that it actually is a commonality, not an orthogonality.
Is the orthogonality of the ability to replicate and proliferate.
Why would you call it? Why do you call it? Sorry. Why
can I just answer the question?
No, just say the word.
There are two things. Yeah, but there are two things in an orthogonality statement. And if you ask me what it is, then I have to say two things before you interrupt, because otherwise I can't explain what I mean. The two things that are orthogonal is the one, again, is the ability to replicate, and the other is the additional functions that the entity might have. And so I'm looking at your description of the process of maintaining identity, in which you nicely indicated the possible route by which, also in the simulations, the composition of the micelles can, under certain compositions, stay the same during growth. And this is an important property in order to ensure the proliferation. And the issue is whether or not, when you make a variant which you want to inherit, whether or not that variant automatically has the same property or not of being able to maintain this information flow of material so that it will also preserve its composition. Because it's a bit like Martin's question in the sense that if the natural tendency is just to go back to the attractor, then mutations and variations will just be lost because you'll go back to what you had before. Whereas what you need is that there's an orthogonality. So when you make a variation change, that variant can also preferentially proliferate itself and not the thing that it came from. So that's an orthogonal stabilization of things. And I'm not seeing in your description, when you describe the model, I'm not saying it's not there, but I'm not seeing an analysis of how that orthogonality comes about.
Thank you for the question. I thought I understood orthogonality right from the beginning, and I'm sorry for my interruption because I just wanted to make sure that I understand it, because otherwise the rest of the question might have been illegible to me. But I think I understand what you're saying. I actually almost sure I understand, and I thank you for the question. Let me try to explain in my own words. The idea is that this is actually a weakness of a model in which there is a sequence, as we know it today, when try to go back very, very long ago to very much more primitive systems. And let me explain that. DNA and RNA are made in such a way that they are completely ignorant of what sequence is being replicated. And it took a long, long evolution to get to this point, because of course, if you want to be as good as DNA and RNA, you want the replicase to ignore the sequence to a very large degree, of course, not to an absolute degree. And this is, if I am not mistaken, this is what you call orthogonality.
That's correct. That's an example of the orthogonality in the RNA case. Yeah.
Thank you, John. I really appreciate it. So what happens is the following. Because you start very clumsy and you start very primitive, there is no mechanism that will ignore the composition. Different compositions will breed through in different rates, different capacities, different exactitude, okay? And this is what gives rise to let me go back. What happens oftentimes with people that go in the footsteps of Eigen, and he is a grandfather of mine academically, because my PhD advisor was his postdoc, Israel Pecht. But the point is that when you come to Eigen's equations, there is no phenotype. So there is no way of saying what happens. So people invent phenotypes, for example, the folding of the RNA. It's nice and well. If you do folding of RNA as an example of phenotype, it means in order for phenotype to survive and propagate the genotype that it sits on, they have to talk to each other. What is so nice about the model, the GARD model, is that it does so naturally. If you stray too much, it won't replicate. But if you make a small mutation, sometimes it replicates better and sometimes it replicates worse. And as written in many of my papers, in the GARD model, the phenotype and the genotype are the same. It's a collection of molecules that interact so as to keep homeostasis, which means reproduction, but also, and they do so by catalytic effects. But the phenotype is how well this happens. So survival of the fittest sits exactly on this non-orthogonality.
Yeah, I understand that point, but the problem is if mutation and variation are fundamentally and violently antagonistic to proliferation, then we have to ask the question. That's not the case with RNA, where the ability to replicate and be replicated is not necessarily different if you get different properties of the RNA because of the common mechanism. But if they are antagonistic, and so that the mechanism by which you stay the same is pulling back to the same composition, then you have to ask the question, what is the mechanism for allowing variation against the force of returning to the same attractor? And so that needs to be analyzed. I'm not saying that you can't do it. I'm just saying that it needs to be analyzed.
We have analyzed it. We have a paper from 2007 in Journal of Molecular Evolution in which we deplete one of the compounds, and some of the compotypes or composomes. Compotypes is just a group of chromosomes, a cluster. Compotypes die, and in others they survive, and in several cases they get better. So I would be very, very happy, John, to speak more about this, because it's a profound question. It's a profound difference between the classical origin of life that sits on the idea that we already have perfect replication of polynucleotides. This is the orthogonality. And between that and what we suggest life began with. Okay? But I'm really grateful for this question.
Okay, thank you very much, and I'll have a look at the 2007 paper in the slide.
Okay. But just maybe one more sentence. You don't have even to go there, because we know that the composition roams around. This is in this slide that you see on the screen now. It's not in one point. It roams around, but it gets attracted here and there, and it ends up getting out. And if there are two composomes, then it may be attracted to another one. So this is another paper from 2014 about the ecology of GARD with first author Markovich, Omer Markovich. Okay. Anyway, we should discuss. I really would like to go forward on that.
Yes, thank you both, because I think this is great. This is the reason why we're doing this, to promote this kind of discussion, which I love it. But I'm sure that there will be other occasions in the future to do this, to resume this discussion. There is another question by Geronimo. Please, Geronimo. Momi, yes?
Yes.
Hi, Momi.
Can you hear me?
Yes, please.
Okay, I'm not sure that my question fits with your seminar, but anyway, you know that there is a lot of work on how cells communicate each other. And
you said horses?
Sorry?
Cavalli?
No, I'm Momi Lanfranchi, my name.
What?
The question
No, I said that there is a lot of work on how cells communicate each other. And the normal way is that they communicate through exosomal vesicles. But okay, but there is also a lot of debate on this issue because many scientists, most of the scientists are saying that this is not a right communication because these vesicles are coming just from the composition of dead cells. So based on your trust on lipids, probably an answer to this question to distinguish between right and wrong messages would be look at the lipid composition of these vesicles. So if you want to, if a cell want to communicate through a microRNA, then the lipid composition of the vesicle should such a way to link this microRNA within the cell and then to send it to another cell. What do you think about it?
So you think maybe in an exosome, the outer lipid, the layer of the outside of layer of the lipid could be some kind of a signature for communication, yes, or for deciding, or maybe they actually carry the RNA inside. So you're asking whether it could be that certain compositions of lipid will prefer certain RNAs and not others.
But they normally do this. You can find a lot of vesicles containing anywhere, anything, proteins, microRNAs, long non-coding RNAs. The question is, are they really a communication or just vesicles that are coming from the composition of dead cells? So no way to communicate, just a signal of dying. So
but if you assume, that you said that maybe it just represents the composition inside, say, of the yes, transcriptome of that cell at this point. But this could also be information for communication because the other cell, receptive cell, could have a different composition of RNA. But to that point, whether what is nice about your question is that we have accumulated a lot of literature on how lipid composition is different in different organelles, including in different kinds of vesicles. And for this caveolin was quite an interesting case in point because it will form the vesicle only if there is a certain composition of lipids at a given domain in the lipid membrane. Mm-hmm. So I would not dismiss the idea that composition of lipids plays a certain role in this presumed communication by exosomes.
Okay, thank you.
Okay, I guess that we have very lively discussions, which I don't see any other. Are there any other questions?
I also don't see that there is time left.
Yes, I know that. That's why we have a meeting now. So we should close this. So let me, on behalf of the whole community, let me thank Doron again. Thank you for being with us, and I hope to see you in the next talk of this series.
Thank you very, very much.
Thank you very much.